Hello everyone. At this point in time, we'll be learning about the structure of DNA and RNA and its organization. Now, DNA, as we know, is considered as a macromolecule and they are very long molecule and they carry our genetic information or they contain the blueprint of life. Like they determine how we grow, how we develop, our mode of reproduction, and even our death. No? So sila ang magdetermina na. And the DNA, they are arranged at 3 levels of increasing complexity. We have the primary, the secondary, and the tertiary structures of DNA. And so if we're looking at the primary structure of DNA, we're looking at, or basically we are considering the nucleotides. Because ang nucleotides manggod siya gyud ang basic nga building block sa tuang DNA. And without nucleotides, dili pod ma-form ang DNA. And these nucleotides are also joined together with one another by the help of this phosphodiester linkage or a phosphodiester bond. And so if nucleotides are joined together with the same nucleotide, like kaparehas niya, then it will become a polynucleotide. Now, for the secondary structure naman of the DNA, it refers to the double helix, no? Muna siya ang katong three-dimensional configuration of the DNA, which is the helical structure. Katong duha ka polynucleotide strand nga gitwist, which was the model that was suggested by James Watson and Francis Crick, the double helix. So that's an example of the secondary structure of DNA. And then for the tertiary structure of DNA naman, basically, it is the higher level structure na, and it is formed by additional twisting, like grabe nga pag-twist, like more twisting, dili lang kaisa nga twist ang DNA, and the coiling of the double helix. Okay? So, the tertiary structure, it is manifested as the supercoiled structure of the DNA. Katunag yung grabe nga pag-twist and then gikumpol-kumpol na sila, like na supercoiled naman sila. So yun, that's the tertiary structure of DNA. And now, let us focus on first the primary structure of DNA. So kailangan sa nga makasabot sa taon sa dining mga primary structure of DNA before ta mo adto sa more macroscopic version of how the DNA are arranged inside our cell. Now, ang kining primary structure of DNA class, mo ni sila ang mga structure nga makonsider nato nga building block gyud. Nga without them, dili ma-forma si double helix or ang secondary structure, the tertiary structure, and even the quaternary structure. Now, the primary structure of DNA class are the following. We have the nucleotides and we have the polynucleotides. So, lahi si polynucleotides and lahi si nucleotides and si polynucleotides, because when you say nucleotides alone, this is the basic building block gyud, from which the nucleotide is made up of 3 major components: the phosphate group, the pentose sugar, and the nitrogenous base. Meanwhile, for the polynucleotides, from the word poly, this is made up of many nucleotides that are joined together by a phosphodiester bond. So, diba, here we have an example, or we have a representation to show the difference between nucleotides and the polynucleotide. So, kining inyong nakita nga nasa box, mo ni siya ang nucleotide. And if we are focusing on the nucleotide alone, ang ato lang yung tanawon anak ay ang phosphate group, ang sugar, and ang nitrogenous base. Now, in this particular example, makita nato nga na ay daghan na mga nucleotide nga naka-join with one another. So, ang DNA nato class, to be specific, is made up of 2 polynucleotide. Dili lang nucleotide ang gakompose sa DNA, especially if we are looking in a bigger scale. Kaya DNA manggod cannot survive alone kapag nucleotide lang siya. Pero mogamit siya nucleotide as its basic building block. So in this case, we have double-stranded DNA. And as we can see here, this polynucleotide strand of DNA on the left side is made up of how many nucleotide man? We have 1, 2, 3, and 4. The same goes on the polynucleotide strand nga complementary pod ato na sa left. It is also made up of 4 nucleotide. Yeah, it is made up of 4 nucleotide. So, ang polynucleotide strand, basically, these are made up of many nucleotides. So, this is the polymer, ang polynucleotide. Tapos, ang nucleotides, mo ni siya ang monomer. Okay. Now, ang nucleotide class, so di ba na-discuss naman nato ni siya? As I have mentioned, the nucleotides are the basic building block of our nucleic acid. So, tanang DNA, tanang RNA, they have nucleotides. And nucleotides are made up of 3 major components. We have nitrogenous base, the pentose sugar, and the phosphate group or the terminal phosphate group. So, terminal siya is because ang phosphate group, pakitaan siya sa tumoy. Naka-attach man guni siya sa ika-5th nga carbon atom sa pentose sugar. Okay. So, we have here. So, nucleotide is made up of phosphate group, phosphorus that is surrounded by an oxygen atom. And then nucleotide is also made up of sugar. That sugar can be in a form of deoxyribose or a ribose sugar depending on the type of nucleic acid. In DNA, the sugar that is present is deoxyribose, and for RNA, the sugar that is present is the ribose sugar. Then, nucleotide is also made up of nitrogenous bases, which are classified as either purines or pyrimidines. So, purines class, they are double ring, which means na siya 6 ka member ring, tapos 9 attach po siya og another 5 nga member ring. So, manag-double ring sila. And then, for the pyrimidine, single ring siya. So, that means na ay 6 ka ring nga naa. Or 6 ka tawag aning, basta na siya sa inyong biochem, kani siya. Basta ang pyrimidine single ring na siya, no? And ang example of purines are adenine and guanine. Tapos, ang examples of pyrimidine are the thymine, which is only present in DNA, the uracil, which is also present in RNA, and the cytosine. Okay. Now, let us learn or let us know more those components that I have mentioned kaya para mas masabtan yun na to. Kaya diba ang nucleotide siya man ang basic nga building block sa itong DNA. And without nucleotide, then wala po tayo DNA. So, ang nucleotide made up of sugar, phosphate group, and the nitrogenous base. Now, I'd like to focus first on the sugar of the nucleotide. So, unsa man nga klase sa sugar ang naa ang atong nucleotide? Now, the sugars of our nucleic acids, may it be DNA or RNA baron, are called as pentose sugar. So, they're called pentose sugar from their pentagon because they have 5 carbon atoms. Na sila 5 ka carbon atoms. Like in this case, so So this is a pentose sugar. We have 5 carbon atoms sila. We have 1, 2, 3, 4, and 5. Yon. Now, a nucleotide contains either a ribose sugar, which is present only in RNA, or a deoxyribose sugar, which is also present only in DNA. So, ang RNA-DI ang tawag sa iyahang pentose sugar is ribose sugar, and then ang DNA naman is a deoxyribose sugar. Nausay kalahian ani nila ma'am nganong managlahi ang ilang pangalan, and in what way na to makita ang ilang difference since both of them are pentose sugar. Now ang RNA class, as we can see, naa siya hydroxyl group nga naka-attach sa ika-second nga carbon atom. Diba? This is the hydroxyl group or the OH group. We have here the first carbon atom and then the second carbon atom. So in RNA, there is an OH group or a hydroxyl group that is attached to its second carbon atom in its pentose sugar or in the ribose sugar. Meanwhile, in the DNA, we can see here that it does not have oxygen on its second carbon atom, the pentose sugar. Instead, we can only see that in the second carbon atom of the pentose sugar, the deoxyribose sugar, there is only a hydrogen element that is attached to its second carbon atom. And so, what is the relevance of this? Unsay kalahian if ang RNA na ay hydroxyl group and then ang DNA wala siya atong hydroxyl group, instead na siya hydrogen alone nga naa sa ika-second carbon atom. Now, it actually has a relevance class if we're looking at its stability because the presence manggod of oxygen atom in the RNA nucleotide makes it more reactive, which means that they are less chemically stable than the DNA. So, in a way, ang oxygen atom nga naa sa RNA, it makes them chemically disadvantaged because they are more reactive and less chemically stable than DNA. Which is why ang DNA is better suited gyud siya to serve as the long-term carrier of genetic information because they are stable. Wala sila atong oxygen atom nga highly reactive. Okay. Now, another major component of nucleotide class are the nitrogenous base. And nitrogenous base are categorized as either purine or pyrimidine. Now, ang purine, di ba, ang purine kaya maumani siya ang double ring. So, this consists of 6-member ring attached to a 5-member ring. And then for the pyrimidine, this is a pyrimidine, it is consists only of a 6-member ring. So for purine, naa siya ay 6-member ring na naka-attach sa naka-attach to sa 5-member ring. So we have here 1, 2, 3, 4, 5, 6, then 7, 8, 9. So this is 5-member ring gyapon kay naman ni duha ka carbon ma-consider ma-apil gyapon ni siya nato sa atong counting. Unlike with the pyrimidine nga 6-member ring gyud siya. Like, dali ra siya ihapon, dali ra siya tanawon ayahang structure. Okay. So the purine and our nitrogenous bases are the adenine and the guanine, and then the pyrimidine are the cytosine, the thymine, and the uracil. Again, as I have mentioned, ang thymine present lang na siya only in the DNA, whereas ang uracil present lang po na siya in the RNA. So, ang DNA, wala sila uracil. The same goes for the RNA. Wala sila thymine. Instead, ang gamiton to pair with the adenine is the uracil. Okay. Now, the nitrogenous bases class also exist in complementary. They are complementary. Like, they follow the concept of complementarity. Like, they complement each other. In which, the purines ng mga nitrogenous base will always bind with the pyrimidine base. So, di ba, ang purines mo itong adenine and guanine. So, kana sila daw, always jud na sila mo bind with its complementary na mga pyrimidine bases, which are the cytosine and thymine. And based on our understanding about the pairing, the base pairing rule for DNA, especially during DNA replication, the adenine will always pair with thymine, thymine will always pair with adenine, and cytosine will always pair with guanine, and guanine will always pair with cytosine. Magina siya ang dapat nga na-establish na, no? Ang adenine, always gina siya mo pares ni thymine and vice versa. Tapos si cytosine, always na siya mo pares kay guanine and vice versa as well. So from here, we can conclude that ang mga purines na mga nitrogenous bases, makig-bind good siya always with the pyrimidines. So, dili pwede nga ang purines makig-bind siya with purine kay dili na nila ma-attain ang complementarity. Like adenine, guanine, or cytosine to thymine baron. So, that's a big no for DNA-based pairing. No? Always good nga kapag purine, makig-bind gyud na siya with a pyrimidine, or pyrimidine will bind to a purine nitrogenous base. Like in this case, as you can see here, di ba makita na to? Si cytosine, ni-bind siya or ni-pair siya with guanine. And ang cytosine is a pyrimidine, tapos si guanine is a purine. And then adenine pairs or binds with thymine. Guanine pairs with cytosine, and then thymine pairs with adenine. So from here, the base pairing rule always apply to purines and pyrimidines. So sila gyud na ang always nga mag-bind. Dili pwede mag-bind si purine to purine or pyrimidine to pyrimidine. So I hope nga nasabta na siya. And also, as we can see here, each of the base pair or katong complementary base pair, they are joined together by hydrogen bonds. So, sa mga molecules class, daghan kayong types of bonds. We have ionic bonds, we also have covalent bonds, hydrogen bonds, mga intermolecular forces. And ang type of bond nga ga-join together sa duha ka base pair is the hydrogen bond. So, kaning hydrogen bond class, weak ni siya nga type of bond. So, dali ra ni siya bungkagon. Muna nga, ang DNA na to, if ma-expose ta og mga mutation causing agent or mga mutagenic agents, mga nang mabungkag gid siya kay ang hydrogen bonds man good, dali ra siya bungkagon or weak nga kind of bond siya. So, the number of hydrogen bonds that can be seen between adenine and thymine are duha lang ka hydrogen bonds. So, ang number of bonds di ay, hydrogen bonds ka makita nato between adenine and thymine, is only 2. We have here 1, 2, and then the guanine and the cytosine base pair are held together by 3 hydrogen bonds. So, makita nato there is a guanine to cytosine nga base pairing na ay tulo ka hydrogen bond nga nag-join sa ilaha. The same goes with the cytosine to guanine. Napoy tulo ka hydrogen bonds nga nag-gunit or nagsumpay sa ilahang duha. Then for thymine to adenine, duha lang. So, constant na gina siya class. Nga kapag adenine, thymine, or thymine to adenine, duha ka hydrogen bonds. And then kapag guanine to cytosine or cytosine to guanine, 3 ka hydrogen bonds. Okay. Now, in a nucleotide also, the nitrogenous base always forms a covalent bond with the first carbon atom of the sugar. In other words, ang mga nitrogenous base di ay, may it be adenine, cytosine, guanine, thymine, or even uracil, will always form a covalent bond and will attach themselves into the first carbon atom of the pentose sugar. No? So, yun. Covalent bond siya. And then, another component of the nucleotide class are the phosphate group. The phosphate, o kabalo na tanagyo na siya phosphorus, and napo siya oxygen. So, ang phosphate group na itong makita sa nucleotide, they are consist of phosphorus atom bonded to 4 oxygen atoms. Okay, we have 1, 2, 3, 4 oxygen atoms. And the phosphate groups, they are found in every nucleotide and frequently they carry a negative charge which makes the DNA acidic. So, di ba, based on my discussion katong sa organization of DNA, ang reason nga nung always nga mo bind sa DNA with a histone is because ang DNA manggod class is slightly acidic, which means they carry a negative charge. And then, ang histones kay slightly positive siya mo na nga mag-bond yun sila or mag-connect yun sila. Nag-strong connection sila because opposite charges attract. Now, the phosphate group is also always bonded to the 5th carbon atom of the sugar in a nucleotide. Alright. Now, always remember class nga ang phosphate group makitaan gyud nato siya in the 5th carbon atom of the pentose sugar. Naa siya sa ikalima nga carbon atom sa pentose sugar. May it be the deoxyribose sugar or the ribose sugar. Naa siya always sa ikalima nga carbon atom. Like in this case, we have this one. This is the first carbon atom, second, third, fourth, fifth. So, ang phosphate group, always nato na siya makita sa ika-5th nga carbon atom sa deoxyribose sugar boron or sa ribose sugar. Okay. So, that's it for the nucleotide. No, so, nucleotide, siya ang basic nga building blocks ato ang DNA. Now, ang atong DNA alone, dili na siya ga-compose or wala na siya all alone na ginusara ng nucleotide lang yun diha sa atong DNA because if we are going to look at the molecular level, daghan gyud kay na mga nucleotide class nga nag-stack up and they form this polymer of the DNA, which is we call that one as the polynucleotide. And so the DNA is made up of many nucleotides that are connected by covalent bonds. So, katulad ang mga nucleotides, they are connected with one another by this type of covalent bond, which is we call that one as the phosphodiester bond. And this covalent bond, the phosphodiester bond, joins the 5' phosphate group, the katong phosphate group na naka-attach sa ika-5th carbon atom of one nucleotide to the 3rd carbon atom nga na naka-attach nga hydroxyl group of the next nucleotide. Kaya magsumpay man na sila. So, kung unsa tong available or katong available nga phosphate group nga naka-attach sa ika-5 nga carbon atom sa isa ka nucleotide, musumpay to siya sa or mo-attach to siya sa ika-3rd nga carbon atom nga naay hydroxyl group sa adjacent pod nga nucleotide. So, nga nang iyang forma para mas masabtan gyud nato. Diba, as we know, ang DNA, they're made up of 2 polynucleotide strands. Since double-stranded man siya, we have here the polynucleotide 1, then we have here another polynucleotide. So, ang DNA class, they run in 5' to 3' direction. So, magsugod siya diri sa 5' to 3' direction. And then, on the other strand, i-antay to niya since they run in opposite direction man. So, ang direction na po sa other strand, other polynucleotide will be 5' to 3', pero ang daga niya is pataas. No? So, if diri sa left side, from 5' to 3', paubos siya, so i-opposite to siya sa next, mo-opposite ning next, or mo-opposite ning other polynucleotide. Iyang anti-parallel manggod sila, so dapat hindi sila pwede nga magkuyog, dapat di sila magkuyog yun. So, mana nga, they will run in opposite direction. So, ang 5' to 3' on the other polynucleotide strand, at banga na siya with a direction nga 5' to 3', pero pataas siya. Okay. So, as we can see here, we have a lot of nucleotides, no? And these nucleotides are attached to another nucleotide through a phosphodiester linkage or a phosphodiester bond. So when we say a phosphodiester bond or phosphodiester linkage, this is the type of bond that connects the phosphate group nga naa sa ika-fifth nga carbon atom, to the hydroxyl group nga naka-attach sa ika-third nga carbon atom of the adjacent nga nucleotides. So, manang masumpay gyud sila kay di ba ang isa ka nucleotide kay nabay na siya ulo nga na siya ulo-ulo niya, which is katong phosphate group. So, katong phosphate group, siya to ang makig-connect with the hydroxyl group nga naa sa adjacent nga nucleotide, which is ang katong hydroxyl group is naka-attach to siya sa ika-third nga carbon atom. And mo ni ang pamaagi by which the nucleotides can join together forming a polynucleotide. Okay? And the strands, the polynucleotide strands, they run in opposite direction, so they are anti-parallel. Dili gyud na sila magkuyog. And then, ang RNA class, they only have one polynucleotide strand, and they will also use a phosphodiester bond in combining their nucleotides, no? In which ang phosphate group makig-friend siya with the hydroxyl group nga naa sa ika-third carbon atom para magsumpay sila. Phosphodiester, like ang phosphate group kay gisumpayan siya og duha ka carbon gani. Duha ka carbon ang ni-attach sa iyaha. Now, the bonds that connect the nucleotides, so balik-balik rin siya, no? Pero ato lang i-expound further kay basin maglisod mag-visualize. Now, the polynucleotides is made up of several nucleotides man, no? And then, these nucleotides, they are connected with the help of this phosphodiester linkage or a phosphodiester bond. And this type of phosphodiester bond are covalent bonds, and covalent bonds class is a form of strong bond. And so, a phosphodiester bond, it is a covalent linkage between the phosphate of one nucleotide. So, we have here. So, for example, we have this 1, 2, 3, we have here 4 nucleotide. So, ang phosphodiester bond, makita nato between the phosphate group here, nga naa, mo ni siyang phosphate group class sa ikaduha nga nucleotide. So, may tabuan ni kay ang kining phosphate of one nucleotide and the hydroxyl group of the adjacent nucleotide, they will join together to the 3 carbon of the deoxyribose sugar in an adjacent molecule. Kaya diba as we know, ang phosphate group naka-attach baya siya sa ika-fifth carbon atom. Tapos ang hydroxyl group naka-attach siya sa ika-third carbon atom. So basically, ang phosphodiester bond from the word di, ang phosphorus there or ang phosphate gipatung-an siya og duha ka bond gani nga ang isa ka bond naa dito sa ika-third carbon atom in which naa dito si hydroxyl group, and then ang isa ka bond pun is naa diri sa ika-fifth carbon atom. And this form the sugar phosphate backbone. So importante kayo klase phosphodiester bond kay siya manggod ang mo sumpay sa tanang sugar, sa tanang phosphate, and from there maka-form ta og backbone nga moay mo protekta sa ato ang mga nitrogenous bases. Okay. So the backbone of the polynucleotide strand is composed of alternating sugars and phosphate group. So ang backbone di ay sa atong polynucleotide strand is made up of alternating sugar and phosphate groups. Pasabutan na agapuli-puli lang sila ba? We have phosphate sugar, phosphate sugar, phosphate sugar. Alternate sila. And then the bases also are projected away from the long axis of the strand. In other words, the nitrogenous bases are hidden in the interior of the polynucleotide strand. So, naa siya makitaan sa interior sa polynucleotide strand. And ang nasa gawas lang kay ang sugar-phosphate backbone, the alternating sugar and the phosphate group. Kaya di ba ang DNA made up of two polynucleotide. And in polynucleotide, we can see there the sugar-phosphate backbone. Okay. Now, an important characteristic of the polynucleotide strand is its direction or polarity. So, always remember, good class, kaya magamit ko niyo siya later on sa DNA replication. Kaya ang polynucleotide strand manggod, they have different directions. They run in opposite direction. And in looking at your polynucleotide strand, we will use this symbol nga 5' end to 3' end. Now, nganong ing-ani man ang ilang pangalan, ma'am? We have 5' end and a 3' end. Now, sige na kong now. So, at one end of the polynucleotide strand class, there is a free phosphate. Na itay free phosphate ka makita, and that is unattached on one side. And this free phosphate is attached to the fifth carbon atom of the sugar, the pentose sugar in the nucleotide. So this end of the strand is referred to as the 5' end. So yung ano yung forma. So here, di ba, we have two polynucleotide strand. Now, in looking at our polynucleotide strand, mag-start kita sa 5' end. And nganong 5' end man, ma'am? It's because 5' end ang iyang pangalan tungod kay na phosphate group nga naka-attach sa ikalima nga carbon atom sa pentose sugar sa DNA or the deoxyribose sugar. Meanwhile, on the other strand, it is referred also as the 3' end. And the reason nga nung gitatawag siya 3' end because it has free hydroxyl group that is attached to the third carbon atom of the sugar. So here, so gitawag po siya og 3' end, it's because na ay hydroxyl group na naka-attach sa ika-third nga carbon atom. So 5' end na ay phosphate group na naka-attach sa ika-fifth carbon atom. We have here fifth, 1, 2, 3, 4, 5. And here in the 3' end, na ay hydroxyl group na naka-attach sa ika-third carbon atom sa deoxyribose sugar. And always remember, class, nga ang direction gyud sa ato ang polynucleotide or sa atong DNA, especially, magamit yun siya nga type of direction or kind of direction later on sa DNA replication is the 5' to 3' direction. So, mag-start ta with that 5' end padulong sa 3' end. So, medyo lisod kayo siya i-internalize, no? Kay ang 5' end naa siya kanang sumpay nga end, when in fact, when we say end, katapusan na siya. Pero diri ade kay diha mag-start. So opposite lahi po dai kapag DNA na ang ato ang tan-awon. We need to cater this another, like we need to expand more our understanding on this. So the direction of our DNA starts from 5' end to 3' end. So mana nga 5' to 3' direction. And our DNA polynucleotide, they run in opposite direction, which is why they are called as anti-parallel. Like mag-antay sila ba, mag-opposite sa ilang direction. Like if we can see here, on the left side, the direction is 5' to 3' end, from top to bottom. So, dili pwede nga on the other strand, top to bottom po siya. Dapat muantay siya ato nga direction nga naa na daan sa left side. And as we can see here, 5' to 3' end siya ang yung direction gapon, pero pa-opposite na siya. So, ang 5' end, na diri sa lubutan, and then pasaka na ang iyahang direction. For moving forward na siya. Dili moving downward. And so, diri apod class, makita na to nga mag-atbang pod gyapon ang direction sa DNA nga. Diri sa other strand, 5' end siya, and then the other polynucleotide strand is 3' end. Then the same goes on the other end of the DNA. 3' end, then ang other one is 5' end. Pero ang direction nila, always remember, is it will start from the 5' to 3' end direction. Then, anti-parallel sila. Alright. Now, DNA also class has secondary structure. So, katong nucleotide, katong polynucleotide, those are the primary structure of the DNA. Now, if we're looking at the secondary structure of DNA, we are focusing on its double helix. The double helical model, no? The double helix. That's it. So, moto katong duha ka polynucleotide nga gitwist na. Nanay twist na anay gi-wound around na siya. Sorry. Alright. Now, the fundamental characteristic of the DNA secondary structure is that it consists of two polynucleotide strands that are wound around each other, like gitwist siya with each other, and it's now called as double helix. And so the sugar phosphate linkages are on the outside of the helix, so naa daw siya sa gawas, and the bases are stacked or projected in the interior, like na siya sulod sa molecule. Mo na nga dili ka kita og base, like sa DNA model, dili ka makakita og bases nga naa sa gawas. Instead, ang naa sa pinaka gawas kay ang sugar phosphate backbone, tapos ang nasa sulod kay ang nitrogenous bases, the complementary bases. Now, the two polynucleotide strands, they run in opposite directions, which is why they are anti-parallel, which means that the 3' end of one strand is opposite to the 3' end of the other strand. So, opposite sila. Okay. Like in this case, we have here a double helix. So, basically, class, if atong i-flatten ning double helix, mga polynucleotide na po ni siya. Duha ka polynucleotide nga gi-twist. No? I-twist around, and the two polynucleotide strands of DNA, they are not identical, but rather they are complementary strand. So, dili gyud sila identical, kay when we say identical strands of DNA manggod, pasabutan na ang adenine diri sa nucleotide dapat amo pares pud na niya kay adenine pud sa other polynucleotide. But then the DNA does not have that type of orientation, and they always follow this concept of complementarity, which is why Ang duha ka polynucleotide strands, they are not identical but complementary strands na sila. They complement each other following the base pairing rule. Ang adenine will always pair with thymine, thymine will always pair with adenine, cytosine will always pair with guanine, and guanine will always pair with cytosine. And they run in opposite direction. The other polynucleotide strand has 5' to 3' direction. The same goes for the other strand, but their orientation is opposite with one another. Okay. Now, daghan kayo tag class sa DNA. We have aDNA, bDNA, cDNA, ddNA, and even the zDNA. And these different forms of DNA, they're also considered as mga secondary structures because they form a double helix. And so let us define what is the difference between the bDNA, the aDNA, and the zDNA. Okay? So let us start with the bDNA structure. Now, ang bDNA structure class, mo ni siya ang klase sa DNA nga gidescribe nilang Watson and Crick. So this is the three-dimensional structure of DNA that was described by Watson and Crick. And the bDNA structure exists when plenty of water surrounds the molecule and there are no unusual base sequences in the DNA. So wala mga kanang mga mutation, wala mga missing, wala mga repetition, wala mga wala na mga DNA. So ang DNA structure, ang bDNA structure, present lang siya when there is plenty of water surrounding the molecule. And another interesting concept also of the bDNA is that they're considered as the most stable form of DNA structure. So siya ang pinaka-stable nga configuration of DNA structure. And as a result of that, this is also the type of DNA nga daghan kaayo nga makita nato within our cell. Their predominant structure in the cell. So that means rampant sila ba? Sila ang naitaas nga percentage nga makita nato within our cell. Kaya sila ang pinaka-most stable form of DNA. So we have here, this is a bDNA, and makita po nato ni mga minor groove and the major groove. So kining minor groove and major groove, because it's more of like distance ni siya sa mga DNA, sa pag-twist gani sa DNA, ana siya. And this also participates in kanang protein gani nga diri mo attach sa mga minor groove and major groove, which has relevance in genetic expression. Now, ang bDNA structure, they are right-handed helix, which means that it has a counter which has a clockwise spiral. Clockwise siya. Here. Clockwise spiral. Clockwise, yeah. Clockwise siya, ana. Dili counterclockwise, but clockwise siya. And then, the bDNA has also 10 base pair. So, ang bDNA class, in every twist ana niya, kay na ay 10 ka base pair nga involved. So, asa man diri ang 10 nga base pair, ma'am? So, we have 1, starting here sa pink, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, manang 10 ka, or I don't know, asa diri mag-start, diri ba sa baba, or diri asa, para ma-appeal ning na sa ubos nga base pair. Pero basically, kapag bDNA structure, naa siya 10 ka base pair per 360-degree rotation of the helix. And so, each base pair is twisted 36 degrees relative to the adjacent bases. So, kini siya, lahi ni siya nga twist of BDNA, then lahi po ni diri sa baba. Or basically, in every 360 rotation of the helix, kinaay 10 ka base pair nga involved. Pwede ito mag-start diri sa baba. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, yun. And then another thing, the base pairs are of 0.34 nanometers ang ilahang kalayon. And so each complete rotation of the molecule encompasses 3.4 nanometer. So this is the complete rotation of the DNA molecule. So they have this 3.4 nanometer nga distance apart with another base pairs of DNA that are also twisted in 360 degrees. And then the diameter of the helix, like ang ilahang circumference in a way, or radius ba na siya? Basta ang ilahang kanang diameter, no? Of the helix is 2 nanometer here. Muna ilahang ang ilang alahi man ang gitasan, pero ang ilang diameter ba? It's more of like katong nasa pa-circle gani ka, like sa imuhang unsa ka dako ba na siya? Ana. And the bases are perpendicular to the long axis of the DNA molecule. And the BDNA has also slim and elongated structure. So pataas ni siya and medyo slim siya type of DNA structure. And then the spiraling of the nucleotide strands creates major. Here we have the major groove. Kini siya, mo ni siya ang major groove. We have narrow siya and deep. And we also have the minor groove in which shallow and dili po kayo siya deep. So this minor groove and the major groove class, they have features that are important for the binding of some proteins that regulate the expression of genetic information. Yun. So that's the BDNA. So always remember nga ang BDNA, this is the type of DNA structure nga prominent sa atong lawas. So daghan kay itag mga BDNA sa atong lawas. And it is also the most stable form of DNA structure, and they're present only in watery environment. Meanwhile, ang aDNA structure, they exist if less water is present. So here, this is the aDNA, and then we have here the bDNA. Now, the same with the bDNA, the aDNA is also right-handed helix, but it is shorter. So, mas mubo siya and wider. Medyo tambok siya as compared with that of the bDNA. And its bases are tilted away from the main axis of the molecule. So, medyo taas. Ang taas sa ila, kay kini sa bDNA man, iba mo ni siya ang long and then elongated, tapos slim siya as compared sa eDNA. Pero the same sila nga right-handed. It's just that for aDNA, they are a bit shorter and wider. And then the aDNA has also been detected in some DNA protein complexes and in spores of bacteria. So yan, that's the aDNA. Present sila if there is less water, and they're also right-handed, but a bit shorter and wider than the bDNA. Now we also have here this zDNA. So we also have that form or kind of DNA. So from the word itself, class, ang iyang pangalan kay nakuha po niya mismo sa iyang own nga characteristic or feature ba. Kay they have this zigzag nga zigzag nga sugar phosphate backbone. So opposite ni si zDNA sa aDNA and bDNA because ang zDNA man good, they are left-handed helix. So here, walhon ni siya. Ang iyahang pag-tilt good class kay pawala. And it also has sugar phosphate backbone that zigzags back and forth, giving rise to its name. And the zDNA structure can result if the molecule contains particular base sequences such as stretches of alternating cytosine and guanine. And the researchers have found that the zDNA specific antibodies bind to the regions of the DNA that are being transcribed into RNA, suggesting that the zDNA may play some role in gene expression. So that's the difference between the aDNA, the bDNA, and the zDNA. Both the A and B, they are right-handed helix, meanwhile the zDNA is left-handed. The slim and the elongated type of DNA is the bDNA. Meanwhile, the aDNA is a bit shorter and wider. Tapos, ang zDNA class, they have this zigzag-like sugar phosphate backbone. Tapos, ang bDNA present siya in the watery environment. And then, kapag aDNA present siya in less water. Okay. Now, let us discuss naman class RNA. So, we are done, we have been acquainted with what is DNA, what are the basic structure of DNA, its primary, secondary, and how they're organized. Like, nakasabot na taon sa gitong kailangan na ito ang masabtan about DNA. Now, let us take a look naman class, the RNA. Because RNA, they're also considered as nucleic acid. So, macromolecule po ni siya. It also carries genetic material. And so, ang RNA class, it is a nucleic acid that is similar to DNA, but with only a single helical strand of bases. So, isa lang ni siya ka strand. So, nag-inusara lang ni siya. Single strand lang ni siya. Unlike with the DNA, nga double-stranded gid sila. Una nga, duha ka polynucleotide ang nag-compose sa DNA. Meanwhile, for the ribonucleic acid or the RNA, isa lang. Siya lang yung isa. Isa lang ka polynucleotide. Now, the RNA plays a key role in turning the instruction that is held in the DNA of our genome into functional proteins in our cell. So, gamit kayo ning RNA class, especially during protein synthesis. Kay dili man gud makagawa sa ato ang DNA sa nucleus. Mao na nga kailangan niyo mo gamit og messenger in which dito na mosulod si RNA. So mao gito iyang role to carry the genetic information or to carry the genetic instruction from DNA so that our body can have a proper instruction as to how or as to what type of protein will be made. And RNA is also made up of nucleotide, which is why it has ribose sugar, a phosphate group, and a nitrogenous base. So its nitrogenous base are the following. We have adenine, guanine, cytosine, and uracil. So it does not have thymine because thymine is explicitly present only in DNA. So it cannot be found in RNA. The same goes with uracil. Uracil can also be found or can only be found in RNA, but not in DNA. Now, we have three major types of RNA. Actually, class daghan kaning classes sa RNA. But in this particular discussion, I will just focus on the three major types of RNA. We have the mRNA or the messenger RNA. We also have the transfer RNA or the tRNA and the rRNA or the ribosomal RNA. So let us define and know what are the functions or what is the function of each of them. Now, the mRNA class is a carrier. So, carrier ni siya of genetic information from DNA to ribosomes. So, siya ang ga-carry sa information or sa genetic instruction as to unsa nga klase of protein ang i-produce sa lawas. So, muna siya ang sulugoon ni DNA, kini si mRNA. And then we also have another type of RNA which is the tRNA. And kini si tRNA class magamit ni siya during translation process in which ang mRNA i-translate na siya into proteins, no? Kay siya manggod ang ga-carry og mga codon, tapos na po ni siya corresponding na anticodon. So kana siya or na siya corresponding na amino acid sa iyang end, sa iyang terminal end. Okay? Then ga-carry po ni siya og anticodon kini diri. Diri ibutang yung anticodon. And then the rRNA, this is the structural unit of ribosome, or this is where protein synthesis happens in the ribosome, or rRNA. So, nani siya large ribosomal subunit and the small ribosomal subunit. So, ma-discuss and ma-learn na to siya further when we discuss now the topic of rRNA. Now, let us go to the difference between DNA and RNA. Like, in what way man maglahi sila DNA and RNA, ma'am? Okay. I don't know if you've seen these memes. But, yeah. So, here, we have, this is how biologists sees the beauty between RNA and DNA in nature. So, we have Game of Thrones that are watched by biologists. So, RNA, isa lang siya ka kulot gud sa buhok. And then, kapag DNA na, duha na siya. So, yun. Alright, so let us compare side by side the difference between the DNA and RNA. So, the complete name of DNA is deoxyribonucleic acid. Tapos, ang RNA, we have ribonucleic acid. Now, the DNA class, they are located in the nucleus of the cell. Meanwhile, the RNA, they're located in the cytoplasm. DNA are also double helix or double-stranded. That means they're made up of 2 polynucleotides. Meanwhile, for the RNA, they are single-stranded, like isa lang polynucleotide ang nag-compose ani nila. And then the type of pentose sugar that are found in the DNA are called as deoxyribose, or they have this type of pentose sugar nga deoxyribose. Meanwhile, for the RNA, their pentose sugar is called as ribose. So they have this ribose sugar. And then the nucleotide bases of the DNA are the following. We have adenine, guanine, thymine, and cytosine. And thymine can only be found in DNA. For the RNA, they have the same nitrogenous bases, nga adenine, guanine, and cytosine with that of the DNA. And what's unique about them is that they have uracil, which cannot be found in the DNA. And for the base pairing, for adenine, they will always pair with thymine. Thymine will always pair with adenine. And then for the cytosine, they will always pair with guanine. And then guanine will always pair with cytosine. Now for the pairing naman, in the DNA, in the RNA, the adenine will always pair with uracil. And then the cytosine will always pair with guanine will always pair with cytosine. And then for the replication, for the propagation, ang DNA class is self-replicating, which means they undergo the process of DNA replication to duplicate itself. Meanwhile, for RNA, they are synthesized from the DNA during protein synthesis, specifically the transcription process, on an as-needed basis. So, depende kung kailangan sa lawas o protein mo nang mag-synthesize ang atong DNA o mRNA. Okay? Alright. So, that ends our discussion for DNA and RNA, its structure and organization. And I do hope that you understand well the difference between the two, because this will be very helpful in navigating the topic of DNA replication and protein synthesis. So thank you so much for your time, and I know that it is a very long video recording, but I do hope na inig ni siyang mahuman and you can learn something from this video recording. Thank you so much for your time.